No, very well. I appreciate everybody sticking around for my presentation with you this afternoon, and I want to thank Karen for the invite. It's a pleasure to be here and chat with you about a disease that I'm passionate about as well. So my plan for this afternoon was to go through and talk to you a bit about Histophilus somni, the bacteria itself. I am not a microbiologist. I'll try to limit my use of acronyms as well, but I do, being vets, we also have our own lingo as well. So one of the cool things, at least from a vet perspective, is the diseases that Histophilus causes. It has many different forms. We will talk about the epidemiology. That's the who, what, where, when, and why, right? Wish I could answer all the how too, but we'll talk about treatment and prevention. So what I'm sharing with you is a bit of what we know, but I'm going to be honest. There's a lot we don't know with this bug yet, I'm sad to say. And maybe like a lot of the topics that were discussed today, that we don't have all the answers yet. If you're like me and you're getting older or you just can't remember your questions till the end, I'm okay if you stop me and we have a discussion as we go. You are welcome, though there should be some time at the end, hopefully, to address your questions, okay? So, to start with, Histophilus somni. It is a bacterium, okay? So it's not a virus. It's and originally, if you've been around long enough like me, or before 2003, it was referred to as Haemophilus somnus, okay? And in 2003, the molecular biologists, right, the gene jockeys, they realized that, hey, Histophilus ovi and Histophilus agni, which are prevalent in sheep and goats or small ruminants, was the same bacteria as Histophilus somni. So they changed Haemophilus to Histophilus, okay, to recognize that. What I wanted to point out was that Histophilus belongs to the family Pasteurelliaceae. Okay, I can't say that 2 times fast, but anyways, realize that Pasteurella maltosida and Mannheimia, those are some of the common bacteria that cause BRD. It belongs to the same family, okay? And that's why we see it show up in BRD as well. The last part of its name, somni, comes from being somnolent or sleepy. And that the first form that was described in the literature relative to cattle was the eye team or brain form. And I'll discuss more of that coming up. So as came out in Simon's talk and others, is that these bacteria are actually commensals, okay? They are normal inhabitants in the upper airway. For Histophilus, it's also in the reproductive tract, which I think is kind of an interesting thing as well. Because you think about some of these calves when they get born, where they're coming through. And so probably the Histophilus that they're carrying in their respiratory tract came from mama cow's reproductive tract, okay? So maybe that's a gross thought, but it shouldn't be. I mean, it's kind of a neat thing. What we know is that there's a lot of strain differences, okay? And these strains have different pathogenicity, okay, or disease-causing ability. And on my next slide, we'll describe some of those virulence factors that they might carry. The other neat thing is these strains tend to go to different parts of the body, and that's why we get the different forms of diseases that we do. So, being a commensal, I also like to think it's an opportunistic bacteria. So when the opportunity comes up and this animal is sick and diseased, that's when, or if it's weakened, it gives it the opportunity to cause disease. And like I said, I'm not a microbiologist, so please bear with me on some of these things. So its surface proteins allow it to bind to both the respiratory epithelia, that's the mucous membranes in the respiratory tract, as well as the endothelia. The endothelial layer is the lining of the blood vessels, okay? And that's going to become more, you'll realize why that's important as we get going here. The other cool thing is some of the surface proteins bind immunoglobulins, and so it kind of prevents the antibodies from binding up the bacteria, and it's able to avoid that in the bloodstream, which is also critical to when it causes disease. The last thing is phase variation. These proteins change the architecture or the structure of the organism, and that allows it to go also undetected when it gets into the bloodstream, okay? Being a gram-negative bacteria, it also produces endotoxin from its outer membrane. The other cool thing is it has co-infections with other bacteria, that it produces bacterial slime, right? I always compare biofilm as being like if you've looked in pipes, maybe you don't want to look in your pipes, but that slimy layer on the outside, that's biofilm, okay? Well, no, but the cool thing is it does that in the pipes of these animals, in the respiratory tract as well as in the blood vessels of the heart and other parts. Sorry, we'll get there. The last bit is free radical inhibition. So a lot of the immune cells gobble up these bacteria and they digest them. And they use free radicals to do that, but this bacteria can survive, okay? The last bit is part of their DNA is in a plasmid form, and they often carry tetracycline resistance. Kind of interesting, eh? All right. So I guess what's really interesting with Histophilus to me is that it causes septicemia. Hopefully everybody brushed their teeth today. If you noticed a little bit of blood on your toothbrush when you were done, some of your plaque bacteria might have got into your bloodstream. Okay, we call that bacteremia. Okay, if you were to get sick or if you were immunocompromised on drugs that maybe suppressed your immune system, maybe some of those bacteria go to your heart and start an infection there. That is what happens in these calves, okay? Not because they brush their teeth, but the point is the bacteria gets into the bloodstream. And how it gets there is when they're having pneumonia, okay? This bacteria is one of the ones that's there active in the infection. And if it gets exposed to the bloodstream and it has different virulence factors that can allow that to happen, it gets access to the bloodstream and starts to circulate to the body. And in these compromised animals, we talked about some of the virulence factors. It allows it to go to different parts of the body in their bloodstream and adhere. And the cool thing is it kills the lining of the blood vessel, and that causes a bleed or oozing, okay? And we'll talk about the different forms, but it's really, I just think it's really neat how it causes its disease. There's been some description in the literature about BRSV. That is one of the common viruses that we vaccinate for. It causes massive lung inflammation. And what it does is it shifts the immune response from being a T helper cell 1 reaction to a T helper 2 reaction. What that means, rather than having cells that gobble up and clean up or mop up the infection, a T helper 2 is an antibody response, okay? The bad part, I told you, they've got these bindings, sites that they can change, so it's really hard for antibodies to clean it up. So it just doesn't work very effectively. The other interesting thing is BRSV causes a massive IgE response. That's what we see in allergies, and if you ever see a set of lungs from an IBR case, a BRSV case, they are an allergic reaction. And so the other thing that we see with histophilosis is sometimes an allergic response that goes along with it. And I had mentioned the biofilm production. Pasteurella maltosida is one of the bacteria that can be found in co-infection and causing some of the biofilm. So it's not just one bacteria that's on its own in that slime, it's multiple that are working together. So the last part I've already kind of, referred to is it damages the lining of the blood vessels and causes the blood clot formation and the disruption of the blood supply. So depending on where it goes in the body, the common sites, as you can read up there, is the heart and the pericardial sac. We've got the brain, the lungs, and the pleura, as well as joints, okay? And some of the diseases, I know as vets, we've got long names there like myocarditis, okay? Myo meaning muscle, card meaning heart, itis, so inflammation of the heart muscle. And the pericarditis is the same, but it's the outside lining or sac, right, that forms around it. With myocarditis, these animals often have labored breathing, shortness of breath, often frothy. These are often pen deaths. When I've looked at some of our data, probably about 50 to 60% of these animals get treated. The other ones just you find them dead in the pen. ITEAM stands for infectious thromboembolic meningoencephalitis, right? The problem is it's not a thromboembolism. Thrombo means blood clot, right? Embolism means circulating through the blood. It's really just a thrombus or a blood clot that forms in the brain. We'll show you some pictures because I'm a vet, right? We have to look at dead things together. So we'll on the next slide, we'll have that, all right? In the lungs, it does play a role in bronchopneumonia or your regular, BRD, but the layer around or the cavity around the lungs is called the pleura, pleural cavity. And we can also have a fibrinous, there's fluid and fibrin or protein that exude into that cavity and make some pretty dramatic lesions that you can see. And putting pressure on the heart and the lungs, they often will show up as chronic pneumonia type animals. We also feel that it goes into circulation and causes inflammation in the joint capsule and will result in fluid getting into the joints and causing arthritis, okay? So lots of different places that this thing likes to go. I made a comment at the bottom, like with blocking the blood vessels, it often results in some effusion. That means some of the fluid from the blood leaking out, so it may not look like an actual bleed, but it will look like serum if you've ever seen that part of blood where it's yellowish, okay? And then the protein fibrin that's associated with it. So, these are some living animals. So this was a myocarditis that Dr. Jansen had provided pictures for. Obviously doesn't look real happy there, but this is pretty typical that you find them acutely, a lot of froth from those animals because they've got so much fluid in their lungs as a result of their heart failure. They can also just look like a BRD, okay, on that top right. And on the bottom is the nervous form. That one maybe looks more sleepy than they sometimes do, right? Oops, sorry, went too far. All right. So like I said, or promised before, we'd look at some pictures from dead animals, or dead cattle. On the top right, I'll move over. That's a picture of an ITEAM. And I apologize. I'm not sure for the people online. Maybe you should have just used you're going to do that. Yeah, thanks. You're awesome. What I wanted to point out there where my putting the pointer, that's the point where there's a bleed within the brain. That's obviously going to cause that part of the brain tissue to not function properly. So the clinical signs that we see with these animals can be quite variable depending on where the bleed within the brain and within the brain stem itself. So that's an ITEAM, right? These are pictures of hearts that have been cut open, okay? These two muscles here are called the papillary muscle. This is the left side of the heart, and it pumps the blood to the body, right? And what ends up happening is the bacteria is lodged in the muscle here, blocks it, and we end up with these acute infarcts, okay? These here are examples of chronic infarcts, okay? So they look more white and scarred. With the blood supply cut off, we get what we call a sequestrum. And what that simply means is there's still heart muscle there, but it's not, it's still got the same structure, it just has no function anymore. And as a result, we get left-sided heart failure with those. And I'll have a better picture from Dr. Jansen later, but this has a little bit of pulmonary edema where each of the lobules are separated by fluid. This here is an example of pericarditis. So this yellow is a thick layer of fibrin on the outside of the heart. They're pretty dramatic. You get something like this with hardware disease, but they really stink, and it's not that nice, clean yellow infection. So this really stands out. And I'm going to jump over to these two here. These are forms of pleuritis. This one's more yellowish looking. I know that's not real scientific, but that's an acute pleuritis. So that's the chest cavity. Our head's over here, back end over here, right? So this is the chest cavity and the heart down here. Everything's covered, right? The last bit, this is some scar tissue. This is a chronic pleuritis. And this is a form that I've been seeing a lot more this last year. We always talk that some of the conditions change. This is the belly of this calf, okay? So not only did the chest cavity have pleuritis, but it also had this beautiful fibrinous peritonitis, we call it, okay? In pigs, they get a disease that's called Glasser's disease or syndrome. And it's actually caused by Haemophilus parasuus. Huh, who would have thought? So it's another Histophilus, like they're in the same family, right, that branched off. I think we're getting Glasser's disease in cattle now, unfortunately. So it's just another form. I think this thing just continues to change no matter what we do. So I didn't show you pictures of arthritis. I apologize. Only so much. Okay. What I wanted to point out, this is just quickly a timeline of when the diseases were first described. It was first isolated in sheep in 1925. The nervous form was in 1956. That's when I-team was first identified. Some of the reproductive stuff through the 60s. And in the 80s, that's when the cardiac and joint or arthritis forms were first described. And then, like I said, in 2003, the taxonomic changes. And it's also been, identified in dairy cattle as causing metritis. So, like I say, that's the reproductive side. Anyways, so just wanted to kind of quickly summarize that. So what do we see in terms of epidemiology? Morbidity I have to question because it's really hard to separate Histophilus, from garden variety BRD. They look very similar when they're sick, okay? What I'm gonna do is over my next couple of slides, I'll take you through a few things that might show, some evidence that I think it's a disease that's more common in the northern latitudes, mostly in calves versus yearlings or adults. We see it in confined cattle, and the larger the feedlot, usually the bigger the issue, okay? Mortality tends to be more associated with cold weather. So what I've got here on this slide is basically case counts for BRD at a feedlot and these are fall placed calves, kind of placed in our traditional October, November, and into December. And what I've got summarized here is on the bottom is the days on feed when these cattle were pulled. And as we've discussed, it's pretty typical that these calves would get Draxxin or a Telithromycin on arrival. So oftentimes, there is a slight delay to when we start pulling and treating. I agree, it probably should be a lot less than what it is, okay? But that's why we start to see this first spike. What I was trying to point out here is you'll notice that this epidemic curve is that we've kind of got a biphasic, okay? The second bunch of pulls often happens at 20 to 20 or 30 days on feed, and we usually see them start dying of Histophilus by about day 30, okay? We can certainly have cases that happen before that, but this is pretty typical for feedlots that have lots of Histophilus, and it's repeatable, I can tell you that. So for the same feedlot, what I did is I just kind of grabbed some of these numbers quick. So looking at their head counts on the bottom per animals per pen, that's on the left side. Oh, I should, for the people online, I should. All right. So we've got head per pen, right? And we've got our Histophilus mortality. That's within the pen. What I want to point out is you can see that some of these pens have significant issues when you're losing 6% of those animals. Not a clear distinction. You know, we have always assumed that we would have more disease with more animals. For this feedlot, it hasn't been clear cut, but I still is a point that we'll bring up and discuss later. Head count doesn't necessarily always relate to density, right? And so those are things that we should probably tease apart better. In terms of BRD or UF or undifferentiated fever, that's what's on the bottom of the right graph, and then the Histophilus mortality. And what I'm trying to point out is your treatment rates don't always correlate to the disease outcome. So, and I think part of the reason is what I said to you before, is that there's lots of animals that don't get treated that end up dying of this condition, that they just die acutely or we treat them too late. All right. Got a couple slides here, so bear with me on, these are mortality curves. And we summarized data from 2014 to 2024. This here only includes the cardiac forms, and the myoc is myocarditis, chronic myocarditis, pericarditis, and then chronic pericarditis. So altogether, and the dark green line at the top, that curve is Canadian beef calves, okay, over that period. And the next hatched line, which you'll see below, is, and oh shoot, maybe I'll keep moving around. All right, might be easier just to stay, right? This line here is US beef calves, and that's my point. There's quite a difference from what we see in Canadian cattle versus US cattle. And these were bought and fed in the US. So this came out of our Telus database. And so we just wanted to compare the calves. As you'll notice, the yearlings, whether they're US or Canadian, these cardiac forms are pretty low. As well as the beef on dairy, okay? So the Holstein crosses, which is kind of an interesting thing to notice. So what is it? Is it genetics? What is it about them that makes, and I can tell you, we've had beef on dairy cross calves, similar weight besides beef calves, and we're having a Histophilus outbreak in one pen and not in the other. So I find it really intriguing to look at that. My last point with this box that I drew on here, like I mentioned to you, the way this curve goes up, it first starts to take off at around 30 days. Certainly there is some mortality that happens before, but they're still dying out at about 150 days, but that is where it tends to plateau. So this slide just kind of breaks them down individually, so the myocarditis and the chronic myocarditis. I think we do a good job of getting these ones treated, and that's why we end up with about twice as many that are chronics, right? The antibiotics does help somewhat, but it doesn't usually keep them alive long term. I'll be honest, I'd love to go to a slaughter plant and look at every heart. I don't think there's many of them that make it to slaughter. I think the ones that get it, they die, either acutely or chronically, but that's Steve's. I had never had a chance to dig into that, but what I wanted to show you is the pericarditis is overall a pretty lower mortality risk of the forms. And on my next slide here is the pleuritis. And what I did think was interesting, this is where the US calves had more than the Canadian, okay? So we beat them in myocarditis, but they beat us with pleuritis. I'm not sure if that's, we won't talk about hockey or baseball, will we? Sorry, that wasn't good. All right. Other forms that I could take you through, but I won't bore you to tears, is diphtheria, endocarditis, septicemia, pulmonary edema. These are all different causes or codes that we associate, at least in calves or certain populations, with histophilus. Okay, so lots of varied things. Okay, what I wanted to show you on this slide is, once again, this was based at December 31st from our Southern Alberta feedlots. I classified those with fall-placed calves as greater than 5,000 or less than 5,000, and these are what the numbers were looking like for BRD mortality, chronic pneumonia, Histophilus, and total mortality. Be interesting to see what it looks like at the end of the year. I can tell you within our Telus database right now, Histophilus is sitting at about 0.7. average. Okay. All right, I apologize. This slide's a little bit busy, but for me, I was trying to think of a way I could show the impacts of weather. Okay, so this is for one of the feedlots I consult with. Basically, from September to December 31st is what our x-axis is. On the left here, we have the temperature in Celsius, okay, and that is the green jagged line, okay, the really wild one. Okay, the blue line there is precipitation, and I don't have a really good axis for that for some reason. I think that was actually supposed to be on the left. Okay, on the right, what I'm showing you is the cumulative percent. Okay, this first hatched line that I'm taking you through is their placements of cattle from 0 to 100%. So they place cattle from about mid-September to the first week of December. That's what this line is. This is the BRD treatments that kind of followed nice and steady through the fall, and then this was their mortality. And truthfully, about a quarter of their mortality had happened by the end of November, and then we've got some nice moisture and it turned cold. And you could just see the change in the calves, and we started having calves drop like nothing with. So a lot of this mortality at this feedlot was Histophilus. And I'm not trying to say, I wish I should have done a better job of presenting it, but I do think that I also understand this is highly confounded data, meaning that there's a lot of things happening at this time. But we expect my mortality to go up to 100% for good math, but so it did have to happen, but it's interesting that there wasn't more earlier. We had a really nice fall until it turned crappy in December, is what I was trying to show you. So that was the epidemiology. My next area, I guess, is treatment. So there is lots of drugs or antibiotics that are labeled for Histophilus somni, and I've got them listed up there, so I'm not going to read them off. I think in terms of treatment efficacy, probably treating earlier is better than later. I admit a lot of people have relied on fluorophenical over the years for treatment of BRD, as a first pull, and I think it does have impact on Histophilus. But like I say, there's a lot of animals that do progress to some of these terminal forms pretty rapidly. The crappy thing is, like I mentioned to you before, biofilm protects it. So if they've gotten to the heart or other parts of the body, it's really hard to penetrate the bacteria because it's not exposed. Okay. So that's one of the challenges we face, and maybe why treatment isn't curative in many of them. I think it delays the inevitable. So talking about, prevention. Where I wanted to start was talking about vaccination. There are some commercial Bactrians. Most of them are combined with the Clostridial vaccine. So I've got a few pictured there. But these were all based on strains that were collected in the 80s. Okay. And I can say this, if you're, from the 80s, it's getting dated, right? That's how I feel anyways, right? I'm from the 70s, so anyways, all good. But point is, they haven't been updated. And so I think that's what we're really needing, is to collect some new strains and get some of these vaccines updated. Once again, having said that, vaccination is challenged by the biofilms themselves that also help, or that block the
the antibodies. This was the picture that I was talking about. If you've seen
normal lungs, right? Better point to it here. They would be nice and pink. These are pretty typical of a classical myocarditis or chronic myocarditis because they just fill up with lungs. They're just wet and heavy. And when you look in the windpipe, it's just full of white froth like you saw in that picture of that animal, okay? All right, so what about prevention and antimicrobials? We use metaphylaxis as we discussed, right? Lots of animals get macrolides on arrival, but truthfully, and for preventing Histophilus, I think it does diddly, okay? No, it's sad. I think it maybe helps with our BRD cases, but the duration of effect is getting shorter and shorter. But it's interesting. We use in-feed, antimicrobials like chlortetracycline. These were initially licensed for use at, continuous low dose for 56 days. I would say more and more recently, there is evidence to show that pulsing at higher doses and for short durations with rest periods is probably more effective. Okay, and most cattle would probably get one or two or a couple of pulses, right? And it might be repeated if they need it. Okay. Here again, I've always felt that it reduces our BRD pulls during that period of pulsing. I'm not totally convinced it stops mortality. And knowing the TET resistance, maybe that makes sense. Maybe we're controlling some of the BRD pathogens, but I'm not totally sure it's stopping Histophilus. Not saying it makes it worse, but I haven't got the feeling that it really is totally improving our situation. And I'd almost have to echo the last part. So the last drug is tilmycosin or Pulmotil. That is also a macrolide that can be put in the feed if you have high morbidity. You have to feed it for 14 days at the rate that's listed there. That's what's on the label. The challenge I know people are experimenting and playing around, but we've already given a macrolide on arrival. I'm not sure that giving more macrolides to these animals is really going to make our problem better. So it's not something that I've used extensively. I know that there is some trial work going on to try and look at that. So don't boo. I mean, for me, the biggest thing I wanted to come back to was trying to reduce stress. I mean, it's the same discussion we have all the time when we talk about BRD. Stress is a trigger for septicemia. You can't have septicemia happen without that, right? The immune system has to be compromised. So to me, we have to go back and look at our management practices, how the calves are weaned, how far we travel, the distance, right? I've heard, Craig and I, we've talked about water at some of these auction marts. I'm sure there's places you don't want to buy cattle. Using low stress cattle handling, bedding. You can see I like bedding, but I think it's important too for these cattle to keep them comfortable. I did show you a little bit of information there regarding pen head counts. We have data that would show on the BRD front, the more cattle that we put in our pens, the higher the BRD risk. I can't help it not feel that ultimately that is what's going on. Since about 2018, we've been seeing an uptick in both BRD morbidity and mortality and Histophilus. When we look at our database, what have we been doing in the industry? We've been stacking more cattle per pen. And why? Part of it in the South, because I helped promote it, was RCC. I loved the stuff to death, right? But I think by putting more cattle in pens, we create some of these issues ourselves, right? And there's got to be some cost benefit looked at in terms of that, but that is something that I'm worried about, okay? Other things as we've put more cattle into pens that I've talked with customers about is water availability. I was really trying to dig out the study. I cannot find it for the life of me, and it's ticking me off. There was a study that clearly showed that if you had 2 waterers versus 1, the infection risk was almost cut in half, which kind of makes some logic. we are creatures of habit. We like to go water and drink, at convenient spots, right? If you had a couple places set up, I am sure I would go to the one closest to me, right? Cattle probably do the same thing. And so we just know by nature that cuts our infection risk. Okay. Biosecurity, I think pen fill time, mixing. I've got facilities where we've got processing, so new cattle coming in and they're treating their sickies side by side. I can't help but not worry from a biosecurity standpoint that that's a no-no, right? Do I have evidence to prove that to you? No, I'm sorry, I don't, but I just can't help but not worry that that's part of it. As our feedlots get bigger, I've got places where I worry, like distance to treatment. Some of these animals are pretty compromised, and you can't help but not think we're putting more stress on them if they're having to walk half a mile or a mile to treatment. So just some things to think about. I did list up here, yes, there's other products coming on the market, the pheromones, probiotics or yeast. I appreciated the presentations here. Trace mineral supplements as ancillaries. They have no label claim for Histophilus, and I'm not about to stand up here and tell you that everybody should start using these. Do I think that there might be populations that could benefit them? Maybe, yeah. I don't want to say they're not useful. I don't know if we've figured out. I don't see them as a blanket fix for any of this, but they might work for certain groups, okay? So I think that's where we're still trying to learn and figure that out. So next step, I think for me, is understanding the variability and the strains within our pens and feedlots. Some of the preliminary work that we've done looks like it's very diverse, okay? We are in the process of developing a better vaccine. So Joyce Van Donkersgoed is leading the charge through a project with Vito, and they're working on developing a subunit vaccine. And many of the feedlot practices have been putting or providing samples or isolates to that project. I will mention, so one of the things that I worked with a customer was to develop an autogenous vaccine in the interim. And there is discussion that within the Vito group, we might do the same thing. The subunit vaccine is going to take a little while to get built. And because of that delay, there is a talk of working together jointly across Western Canada across the vet practices is to build something that we could all share and utilize. Right now, I've got one for a customer, but we have to send to the US to get it made, and it's a real hassle with the CFIA, as we were discussing this morning. So bringing vaccines in for one customer and then customizing it for every single client makes no sense to me, right? I'd rather have something a little more generic. So you'll be hearing more that, yes, I think there is some advantages to updating vaccines. I can tell you that with our autogenous vaccine, yeah, we've had some success. It doesn't cure all, okay? We're still seeing disease, but it's certainly minimized it from what it was. I think just understanding some of the co-infections with the other organisms, so some of the other projects going on, whether you're looking at microbiomes or some of the work that Simon's doing is invaluable. So I'll summarize really quickly, right? So Histophilus is a commensal bacterium capable of causing very severe systemic disease, okay, with lots of different disease manifestations. It is interesting to me, the cold climate, why it's a Canadian issue or northern latitudes. But we do see it every year in our young calves in confinement. Like I say, treatment's not very effective. I think preventative strategies, I think we're going to have to start rethinking these. I mean, I appreciate we're doing the best that we can do, but hopefully we can come back in a few years and have developed some new ideas of other ways, and maybe it is vaccines or other things that come on the market. So with that, I appreciate everybody sticking around. I know it's been a long day, so if you just want to get out of here and you don't want to talk to me more right now in front of everybody, I'll stick around for a bit if you do want to chat. So thank you very much.
So So Steve, actually, I was driving down from Calgary yesterday and listening to Bovine Science with BCI, as one does, and they mentioned that water paper. So yeah, that was one of the episodes that I listened to. My question, so do we know how often biofilm formation happens? That's the first part. Does it happen all the time or some of the time?
Yeah, I would say that's all the time.
So can we find a way to bust the biofilms?
That's a great question. And I'm not a microbiologist, but I'm sure. Oh, just like I'm trying, thanks. That's a great question, and because I could have, yeah, they should have heard it online. Okay.
I had the mic.
I'm not sure if there's somebody else.
Bob is a microbiologist.
Aha, there we go. Thank you.
Well, I'm here to ask a question, not answer a question. That was really fascinating. I wasn't aware of this. I'd heard of it in terms of something you see in BRD samples, but anyway, I'm curious, have they looked at differences between the isolates that you get from, say, the heart versus the sac of the heart versus the brain, to see, like, are those all genomically similar, or are you getting different serotypes targeting different parts of the animal?
Yeah, so great question. Part of the reason I chose to send my isolates or samples to the US was they were doing sequencing to try and separate them or look at their commonality, right? And what we ended up finding was that I had two different strains going on in the feedlot that we were building the vaccine for. So we ended up picking two different, even though there was about 20 isolates, they kind of fell into those two groups. This past year, though, with our sampling, we found a third strain. So it's kind of, yeah. So we've picked out another one to add to the vaccine for this coming year, right? And that's how I'm trying to approach it. As far as knowing serotype or the specific isolate type, I haven't had them characterized to that extreme to know, how they would, in your microbiology database, how they would fit into the references. I guess I don't know how they fit there, but certainly we are trying to look at that. And I think as far as the VITO project is doing the exact same thing, so we're looking at where we source them from in the body and trying to characterize that. But there's a lot of variation there from what I've gathered. And Joyce is probably still on there, and she's probably screaming at me right now, but maybe she'll put it in there. She can answer too better than I can, but yeah.
Can you isolate it from the environment, or just?
Yeah, so this organism is not something that lives in the environment. I've been worried about that, and we have done some testing in water bowls and other ways, because I was worried for some of our feedlots, this is a every year problem, right? And you're like, well, we must have something residing within the cattle or within the environment. But no, there's no good documentation. It's pretty fastidious that way. It doesn't survive well in the environment. So, yeah,
Sorry, I have 3 for online. Flavio is wondering, what is the difference and how to identify arthritis and mycoplasm when developed in the joints?
Yeah, so I think arthritis due to mycoplasma is we often still look in the lungs for the typical caseous necrosis. I apologize, those are feta cheese-like abscesses in the lungs, okay? But so oftentimes you don't have arthritis without having some form of pneumonia with mycoplasma, as compared to with Histophilus, it might entirely just be multiple joints that you'll have this yellow serous fluid, and often there's flecks of fibrin. It's usually not as formed as what mycoplasma is. They do look a little different grossly within the joint fluid, sorry.
And Dr. Joyce asked, is the increase in peritonitis that you have seen in all the feedlots or only a few? And did you culture HS from lesions, and were there other signs of HS in these cattle?
Okay, so to answer that, yes, all these cases have had usually pleuritis and maybe some pericarditis, and it's usually chronic. And it's kind of neat because it's along the diaphragm, and you can just see it radiating through the diaphragm, all the swelling. You think of the diaphragm's just a muscle, right? And that infection seems to almost looks like it's just exuded. So yes, they have other signs of HS. That's why I'm suspicious. Now, the kicker is I haven't been able to culture it, and it ticks me off. But these animals have all been, usually they're noted with these big bellies, and they get treated as a peritonitis. So they get usually multiple days of treatment. They've all had antibiotics, the cases that I've looked at, and so I haven't been able to isolate it to really confirm my suspicion. And it's a chronic condition usually, so I think that's the hard part. Sorry.
And one last question from Cam. Do calves vaccinated on the ranch respond better to treatment? I also have comments from Dr. Joyce, but I don't know if you want me to read those.
Okay. Well, I don't know if I do either, actually. Maybe we'll review those. No, if you think they'd be productive, I'm not offended. You're welcome. But if not, we can. To answer, that last question was, ranch calves. Yes. So the kicker is using the commercial vaccines that I discussed. Lots of ranch calves get that, and honestly, I don't think it makes a hill of beans difference. Now, truthfully, maybe it helps with the I-team form. We don't see large numbers. Like when I was a teenager growing up, that was what I got used to seeing, right? Was the I-team form of it. That was my first exposure to this disease. But we don't see as much of that as we used to. So I think maybe that vaccine does actually work for that form. It's just not covering any of the other parts of the body. So.
Do you think we're targeting, whether it's vaccines or drugs like Duraxin, we're using them at the wrong times, or CTC? I just think when these calves come in on arrival, they're already so stressed. And then we hit them with a couple vaccines, we hit them with Duraxin, then a lot of the times they haven't seen silage, or there's usually high hay content in the ration, but we're exposing them to all these new things, even though maybe vaccines shouldn't be new to them. And I think their immune systems are so overloaded, like they don't know what to respond to. So I guess my question is, do you think we need to reevaluate when we're giving certain things and when we're using certain tools?
Yeah, so I mean, people have looked at delayed vaccination, pushing that to 2 weeks or a month after arrival. It's always a struggle. I agree, I understand what you're saying. At the end of the day, it still always has turned out health-wise that you're beneficial to go ahead and do it at arrival. We know that it's not perfect, but delaying it till 30 days, the horse is already out of the barn. So I'll even comment on my autogenous vaccine. So the first fall we used it, we ran short because I only bought a little bit because I just wanted to try it. Everybody had scared the bejeebies out of me that I was going to kill cattle with it. Well, no, but it is a bit of a boogeyman, and I respect that. I've always liked using commercial vaccines because you have somebody else. It's been more tested and proven, right? But going down the road of this, so we ran short. What we ended up doing is we vaccinated these calves at 21 days, and they had terrible outcomes. And I think trying to do that then, they were stressed. I think they already had HS at that point, and it made no difference. This year I played around with it, and we did every other pen at 21 days just for a booster, and it made no benefit. So I would never go and booster this HS vaccine again. That's what I've learned. It's got to be given at arrival. So while I understand what you're saying, I respect that, like the stress they're under and all that. And I really wish it wasn't the first time some of them were seeing vaccines. You're right, that is a challenge. Yeah, I don't want to go on about preconditioning because no, right? But I do see that, right? Like they're the ones that do really well a lot of times.